Okay, this is chapter 6, section 2, and it's called the process of meiosis. And the text for this section is that students recognize the significance of meiosis to sexual reproduction. And the key concept here is that during meiosis, diploid cells undergo two cell divisions that result in haploid cells. And to remind you that this is an egg right here. And then these things right here are the sperm, where only one of these, some 100 sperm is going to successfully fertilize this egg. And then I showed you these two pictures in the last section where the DNA, how it's contained in cells, is within these chromosomes. And the reason for that is because DNA, when it's completely unwound, it's actually very, very long, and it won't be able to fit inside this nucleus. So we wrap the DNA around these little orange thing called histones in order for it to fit inside the nucleus. And then within the DNA, you have these little segments called genes. And one gene, so each gene codes for a protein. You can think of the whole entire DNA strand as like a cookbook where all these different genes, say you have a gene here, the gene here, here, and so forth, where each gene codes for a specific protein. And then you have adenine A, thymine T that go together, and cytosine C goes with guanine G. And then this right here is what's called your sugar phosphate backbone. And this, you can kind of think of the sugar phosphate backbone as like the binding of a book. And then the nucleotides, or the A, T, C's, and G's, is sort of the pages of the book. Okay, and then this is a human karyotype where there's 23, so from 1 to the final pair, the X and Y, 23 pairs of human chromosomes, where these are referred to as a homologous pair. So a homologous pair, these colors in this picture represent genes, where this right here and this right here represent the same gene. The code of that gene might be slightly different, but it's going to be the same gene. Whereas the genes on chromosome 2 right here are going to be different from the ones on 3, 4, 5, etc. And then the XY represents male for these two sex chromosomes, and the XX represents a female. Okay, and during meiosis, you go through 2 rounds of division. Whereas if you remember during mitosis, you only went through 1 singular round of division. And really the two main ideas that you're trying to do with meiosis, you're trying to reduce the chromosome number so that you can bring together the chromosomes of a sperm and an egg and still have the proper number of chromosomes. And then you're trying to create genetic diversity. So genetic diversity meaning that each of these cells that are produced from this one original cell are genetically unique. And they result in haploid cells. So in the previous slide I said there's 23 chromosomes. And this 23 we call an N, where a haploid cell is referred to as N, where they only have one pair, so only one of each of the 23 chromosomes, whereas your body cells, or your somatic cells, so things like your skin cells, your gut cells, literally any cell except for your gametes, or the eggs for the female and the sperm for the males, have what are called diploid cells, which the diploid, di meaning 2, are 2N. So they have 2 times the 23, so 2 pairs of each chromosomes. So they have 46 total chromosomes. Another thing with meiosis is that it only takes place at certain times in an organism's life cycle. For females make all of the eggs that they're ever going to have while they're developing as an embryo inside their mom, before birth's been given. Whereas for males, they more or less continuously are going to keep making sperm after puberty. And in meiosis, the purpose of this is for sexual reproduction, to produce the gametes, or the eggs for the female and the sperm for the male. Okay, so the first phase of meiosis is called Meiosis I, and the second is Meiosis II. And they have 4 phases. And these phases are nearly identical, really identical in concept to those in mitosis. And I'll show you what I mean by that on the next slide, or the next couple slides. And during meiosis I, you're going to split the pairs of homologous chromosomes. So the pairs, so initially, we only have one pair of homologous chromosomes whenever we were talking about your body cells, but whenever you're talking about, anytime you're talking about mitosis, and meiosis, you have to remember that the cell undergoes this DNA synthesis process. So when we talked about mitosis, before we actually got to the actual phases of mitosis, we underwent this process where we replicated the DNA. And the same thing is true when we're talking about meiosis. And again, these homologous chromosomes are similar, but they're not going to be identical. So this chromosome right here is going to be similar, but not completely identical to the one next to it. And the sister chromatids are going to be, sister chromatids being this right here and this right here, are what they're going to be separated during meiosis II. And sister chromatids are going to be exact copies of each other. This is going to be the same as this, and this the same as that. So this is getting into the actual steps of meiosis. So meiosis I, again, it's going to occur after you've replicated your DNA. In meiosis I, you're going to be dividing the homologous chromosomes. But, as I mentioned, the 4 phases of meiosis are the exact same names as those in mitosis, where you have prophase, metaphase, anaphase, telophase, and then finally you have cytokinesis. Where during prophase, you have, this right here is the nucleus, and these little holes right here are representing that the nucleus begins to break down. And then these chromosomes right here begin to condense. And the reason why they condense is because it's easier to separate something when it's really condensed versus when it's more kind of loose. Think about when you're trying to dish out spaghetti. Spaghetti's all loose and kind of stringy, where it's kind of, it slips out of your fork if you try to scoop it out of the pot into your bowl. Whereas if you kind of have it, if you had that spaghetti all nice and wrapped up and tight into kind of like a ball of spaghetti, that'd be much easier to put onto your plate. And then you have these right here are your centrosomes. And then each one of these right here, each one of those things specifically is going to be a centriole. And these are what produce what are called the spindle fibers, these long things in blue. These spindle fibers are made up of microtubules. So if I write it down, microtubules, and these are going to be what in the next phase is metaphase. These attach right at the centromere. Remember, the centromere is what holds these chromosomes together. They hook on at these chromosomes, at the centromeres, so that they can have something to hold on to, so that they can pull them during anaphase. So prophase, you get chromosomes are going to condense, and the homologous chromosomes begin to pair up. So this right here and this right here are a homologous pair, and this right here and this right here are homologous pairs. So they begin to kind of clump together. And then in metaphase, the chromosomes meet up at the equator, right here in the middle of the cell, where you have one of the pairs on one side and one of the pairs on the other side. One side, then the other. And then your centrosomes have migrated completely to the opposite ends of the cell, and then the microtubules have completely elongated out and attached to the centromere. Then in anaphase, you have these the microtubules of the spindle fiber, they begin to shorten. So you kind of see how what's pulling, they shorten, and as they're shortening, they're pulling these homologous chromosomes to opposite ends of the cell. So you could kind of, just for simplicity, they only showed 2 pairs of chromosomes, but, imagine this as chromosome 1 and chromosome 2, and this being chromosome 1 and chromosome 2. Then in telophase, you have the reformation of the nuclear membrane. So the nucleus begins to reform. And then in the spindle fiber, what hooked onto the centromere begins to degrade. And then in cytokinesis, that's when you think cytoplasm. That's where you divide up the cytoplasm. Cytoplasm, which is referring to the watery stuff that's on that all of the materials of the cell float around in. So all of everything that's outside of the nucleus. Okay, and that's meiosis I. So meiosis I, you separated the homologous chromosomes, and then you have meiosis II, where you went from 1 cell in meiosis I to now 2 cells, this one right here and this one right here, and so now you're going to be dividing the sister chromatids. And it's important to note that you're not replicating the DNA between meiosis I and meiosis II. And then it's the exact same idea. You have the same 4 phases, the prophase, metaphase, anaphase, telophase, and cytokinesis that you had in meiosis I and mitosis. And it's the same basic idea. Nuclear membrane breaks down, the chromosomes condense, the spindle fibers and the centrosomes begin to go to the opposite ends of each of the cell, and then you have the chromosomes line up in metaphase at the equator, or the middle of the cell. Centrosomes are on opposite poles of the cell, and then the microtubules are going to hook on right at the centromere. And again, you can think of this as chromosome 1, and then this is chromosome 2, where if they had a bigger picture, you would have 23 different chromosomes instead of just the 1 and 2. Then in anaphase, you break apart the sister chromatids. Sister chromatids, that's a sister, that's a sister. You break these sisters apart by shortening these spindle fibers. And then in telophase, you reform that nuclear membrane right here. And then the spindle fibers break down. And then in cytokinesis, you divide up the cytoplasm again. And then that's it. That is meiosis, where you end up with 1, 2, 3, 4 cells. And each one of these cells is haploid. It's just N. And remember, N refers to 23. So each one of these 4 cells has 23 chromosomes. So let's talk about the differences, similarities and differences between meiosis and mitosis. So meiosis is 2 cell divisions. Mitosis is only 1. Mitosis, you don't have any pairing up of homologous chromosomes. Meiosis, you result in haploid cells, so the N, and then mitosis has the diploid cells, so 2N. Meiosis has genetically unique cells. This cell is unique from this one, from this one, from this one, and there's 4 of them, whereas in mitosis you create only 2 cells, and they're genetically identical cells, and they're diploid. Mitosis takes place throughout the entire organism's lifetime. Meiosis takes place only at certain times. Mitosis could also be involved in asexual reproduction, so whenever you're talking about your prokaryotic cells, so your prokaryotes, so things like your bacteria, whereas meiosis is used for sexual reproduction, where you're making your gametes. Okay, so final slide, and that's just kind of going over some of the specifics of the egg and the sperm. So gametogenesis is referring to the entire process of making the gametes, or making the sperm and the egg. Whereas meiosis is just talking about it. Meiosis is just focusing on nuclear division, or dividing up the DNA. But gametogenesis is referring to the process as a whole. The process where you complete, the complete process of making all the sperm cells, or all the eggs. Interesting thing to note is that when you're making the sperm becomes streamlined and modal. So if you think back to that picture at the very beginning of the PowerPoint, you think of that egg, and then all of those, think of the size of that egg compared to the size of the sperm. Those sperm are really small and really long. And that's because these sperm develop these little tails called flagella. And these flagella, I mean, it's literally like a little, like a little rudder, a little, kind of this, almost like this little fin that the sperm will just wiggle back and forth and kind of use it to swim along throughout the throughout the reproductive tract of the male, and then the reproductive tract of the female. And the sperm also, by streamlining, that means that they get rid of a lot of their cytoplasm and a lot of the other organelles that make them up, and they stock up, they increase their mitochondria. If you remember, the mitochondria are what the term the powerhouse of the cell. They're the ones that are really involved with making energy in the cell. So you want a whole lot of mitochondria because you're doing a whole lot of motion. They're doing a lot of swimming and moving around. So they want to kind of specialize in making that energy, and then stocking up on kind of machinery to power this flagella. And sperm, so when a sperm fuses with the egg, the only thing it really contributes is just going to be the DNA. Whereas the egg cell is what's going to contribute DNA, but not only the DNA, it's also going to contribute the cytoplasm, the organelles, everything else outside of the DNA that we think of when we think of a cell. Another interesting thing is that when you make sperm, you take one cell and you make 4 sperm cells. But for females, when you're making eggs, you only make 1 egg for each cycle of meiosis. And in the process, you make 3 what are called polar bodies. And polar bodies are, they're kind of just useless little cells that just kind of get shot off, and truthfully, I don't think they serve any functional purpose. They kind of just get tossed away. And the reason the body does that is because it wants to stock up on this egg. It basically pretty much puts all of its eggs into one egg, so to speak. It takes kind of all of the cytoplasm and all of the different organelles, kind of everything, and it contributes it all to one singular egg, so that you can kind of have this one egg that's got all of these nutrients and all of these things to help that the embryo, or to help that baby develop once it meets its sperm. Okay, and that's going to do it for 6.2.